Phenology, the timing and cyclical patterning of biological life cycles, is a term many people don't recognize, yet it's a key indicator of global environmental change and plays an important role in structuring ecological communities. As global land use and climatic change have altered environmental cues like temperature and precipitation, species have shifted their phenologies. Take, for example, the earlier spring blooms we've been seeing in the northern hemisphere, or the way that the timing of major bird migrations has changed. These phenological shifts have widespread consequences, impacting not only the individual species changing their phenology, but also all of the other species that they interact with. So although we've become increasingly aware of the ubiquity and relevance of global phenological change, phenology continues to play a crucial yet understudied role in shaping plant communities. Hence, as part of a broader research effort within the University of Oregon's Dietz Lab, we studied the relationship between phenology and species interactions among annual plants. We assessed how neighborhood competition affects the flowering times and fitness of Clarkia purpurea, an annual native prairie forb. To do this, we developed a planting area containing Clarkia and other native prairie species, marked unique focal plants of Clarkia, and collected data about how each focal plant's competitive neighborhood affected its flowering times and seed output. Conducting this research, we found that there is a reciprocal relationship between flowering times and competition. That is to say, not only can differences in species' flowering times influence the outcome of competition between them, but competition can also cause species to undergo shifts in their individual flowering phenology. One of our most important findings was that competitor diversity can influence when plants flower. When Clarkia was competing with a greater number of species, it tended to flower earlier and for a shorter period of time. This is important as it indicates that biodiversity loss could exacerbate phenological shifts associated with global environmental change. We also observed that Clarkia's flowering times and fitness were correlated with one another, meaning that phenological shifts could impact how plant populations grow and change year to year. Overall, our results suggest that the relationship between plant phenology and competition is complex and could influence ecological communities in non-intuitive ways. Therefore, as we move forward with efforts to restore and conserve our plant communities, it is paramount that we continue to consider phenology and use its study as a way to understand, adapt to, and mitigate the effects of global environmental change.